So, good morning, everybody, and thanks to the organization for giving me the opportunity of being here. I'm a CRA, so I've been involved mainly in monitoring clinical trials in pharmaceutical field. And so actually, I'm here just to give you an overview of the state of the art on what clinical trials are both in medical devices and pharmaceuticals. So we will go through different points, so starting from the legislation of clinical trials, and we will see a comparison between the structure of the two and the differences between the clinical trials in pharmaceuticals and in medical devices. So starting from pharmaceutical clinical trials, the legislation, so what actually rules the conduction of a clinical experimentation is the Directive number 20 of 2001, which actually have been repealed now by the Regulation number 536 2014 in order for the conduct of clinical experimentation of human use. And this is in order to create an environment which is more favorable to conducting clinical trials in EU with the highest standard of safety and for participant and transparency on trial information. So this will increase the efficiency of all trials in Europe with greatest benefits for those clinical trials which have been conducted in multiple member states. So which is the best way in conducting clinical trials? We first must consider ethical point of view, and which start from the principles that have their origin in the Declaration of Helsinki, which protects the rights, safety, and well-being of human subject. And so what actually we have to take into account is that all subjects which want to enroll in a clinical experimentation must be free, because no improper influence or inducement have been made to subjects that want to enroll. We have to take into account the compensation and additional healthcare responsibilities and communication with ethics committee, which eventually could evaluate favorable or not favorable in the conduct of a clinical experimentation. And we have to take into account we have to take into account a vulnerable population, and of course the presence of an informed consent must be mandatory. So actually the rights of human subjects have been protected through guidelines which are listed here. So first, the International Conference of Harmonization, which brings together the regulatory authorities of Japan, Europe, and US. The good clinical practice, which define the roles of all the participants of a clinical experimentation, so sponsor, investigators, and monitors. The Declaration of Helsinki, as I said before, which protects the ethical principles of human involved in a clinical experimentation. And the Nuremberg Code, which set up the principle for human experimentation during the Second World War. Now, the clinical research has divided to two kinds of clinical trials, the observational studies and the real clinical trials. So observational studies observe people in normal settings, so it means that research gather information, group volunteers, and according to broad characteristics. And of course, we can have comparison between testing and research, testing and questionnaires or exams that can be given to different patients in order to have more information about what the effects of the differences, lifestyle on cognitive health. And so these studies eventually may be helpful for a possible clinical experimentation that will come. And then the real clinical trials that is used to learn if a new treatment is more effective or less harmful side effect than the standard treatment. So actually we will see the evaluation of a medical, surgical, or behavioral intervention. Well, pharmaceutical clinical trials are well structured and they are subdivided into different phases. So phase 1, which is performed on a small group of healthy people, actually from 2 to 80 people almost, and is aimed to find out how much the drug is safe to give and what are the side effects and how the body gets rid of the drug, so the metabolism, and if the treatment is effective. The phase 2 instead is performed on a larger group of people, let's say in hundreds of people, and in order to find out more about the side effects and how to manage them eventually, more about the best dose to use, and if the new treatment works well enough to be tested in a larger phase 3 trial. The phase 3 here goes from several hundred to 3,000 people eventually enrolled, and the trial aims to find out which treatment works better, more about the side effects, and how the treatment affects people's quality of life, and eventually the standard treatment could be compared to the treatment which is, let's say, experimented with a completely new treatment, different doses of the same treatment, having the same treatment more or less often, or eventually a new way of giving the standard treatment. The phase 4, instead, is a post-marketing phase of clinical trials in order to monitor for a large, diverse population for a longer period of time. So eventually, there is a kind of surveillance system that must collect all the data coming from the reports of side effects eventually, or difficulties in managing the drug, which is called the pharmacovigilance. Well, what we have to consider when we start in conducting a clinical experimentation is the competing factors. So I have listed them in order to give just an idea of what we have to take into account. So coordination of manufacturing activities, clarity of trial protocol and case report form, the ethical and regulatory reviews, site capacity, management and experience, management of different partners, contracting and agreements, trial timeline, which is a very big issue and we will see later, and the parallel activities eventually. Well, we have different kinds of clinical trials, open label, randomized, double-blinded, control or not control. But the randomized clinical trials, since their introduction in the 1930s, have been the gold standard method to assess the clinical efficacy of new therapeutic intervention. And what does it mean, randomization? Randomization means that you have a control group and an experimental group in which the eligible populations in both of the groups have been chosen at random. And now, because of different issues that we will see here, randomized clinical trials, I say, have been challenged. And we will see why. So first, the complexity of trials. Those people running clinical trials are feeling the pressure to design the trial that give the right answer. And the regulation, okay, so many respondents feel constrained by the complexity of the guidelines they follow. Costs, of course, because the trial timeline is putting more pressure on the need for resources to implement the control at every step. So during a clinical experimentation, we have different steps we have to monitor, so we have different checkpoints. And because of them, we will need more personnel involved, and of course, the cost runs higher. So here also the patient access, which is becoming, let's say, trivial because we have high percentage of patients not meeting the targets and dropout rates, which is increasing. The staff and role responsibilities, which are increasing and increasing because of the timeline, as I said before, and because of the expertise of all the personnel involved in a clinical experimentation. And also the technology, which could be the future, but we have to know how to select and use the right technology in order to conduct a clinical experimentation. Governance and oversight, all the people involved, so the strategic partnership, vendors, study sites, CROs, all of them play an important role in almost every step during a clinical experimentation. And of course, new drug with brand new classes of drugs require different ways of running trials because of different kind of areas we know. And we have cell therapies, genomics, and personalized treatment for different kind of population and for different kind of diseases. So actually, this is a summary of what I've said of all the problems during the conduct of a clinical experimentation. So we have getting the right patient is difficult. Difficulties in data collection. We have a lot of data we are collected, but how do we know we use them? And proving clinical effectiveness of new therapeutic intervention. Inefficient trial design, as I said before, the difficulty is to design a very good protocol that could be eventually useful in giving results for the treatment of new therapy, of new disease, or eventually for new therapeutic interventions. So too much trials are not worth doing, and we don't need 100 trials. You may get an answer in 10 if you do it carefully eventually. And of course, the cost, as I said before. The cost of develop, for example, the cost to develop and gain FDA approval for a new drug today is more than $2 billion and drive much of the cost of human clinical trials. Now let's keep on the medtech area. And we know that, you know, of course, better than me, I think, that medical devices involved in a clinical experimentation are used to treat, prevent, or eventually doing screening or diagnostic during a treatment of the disease. So the clinical trials, we have different kind. We know that for pharmaceutical clinical trials, we have well-structured phases. And instead here, we have different kind of clinical trials according to the design of a new prototype. And so we have the early feasibility study, in which the study is performed typically before the device design has been finalized. The first in-human study, in which the study of a device for a specific indication is evaluated for the first time in human subjects. The traditional feasibility study, which commonly used to capture the preliminary information about safety and effectiveness of the device. Pivotal study, which instead is the real clinical study, clinical trial investigation, designed to collect the definitive evidence of the safety and effectiveness of that device. And the post-market study, in order to monitor, as for pharmaceutical area, to monitor whether our medical device is safe and let's say is the usage and eventual associated problems. Well, prior this kind of medical device regulation, the 745/2017, actually in medical device clinical trials, we didn't have a real regulation in order to contact them. But now, because in 2020 it will come into force, it says that at the whereas number 64, the rules on clinical investigation should be in line with well-established international guidance in this field, such as the ISO 14155/2011. And so it says that we have this kind of regulation that says that in order to conduct a clinical experimentation in medical devices, you have to take into account this kind of ISO. And so actually it specifies all the standards and general requirements for all the conduct of a clinical experimentation. And now we will see which are the barriers to put on the market a medical device and so to conduct a clinical experimentation eventually. We have almost 5: the timing of assessment, the acceptability, the learning curve, the choice of a comparator group, and the blinding. So starting from the timing of assessment is a very big issue because we have to consider that a medical device often undergoes frequent changes in device design. And eventually, we have to take into account during your planning that changes should be categorized, such as substantial or not substantial, and to declare them, of course. The acceptability, there's a very big issue, especially in randomization, when a medical device is not chosen by the patient or by providers, and so they refuse randomization. Blinding. For ethical or practical reasons, blinding is often more difficult to perform in randomized clinical trials on medical devices compared to pharmacological clinical trials. And the choice of a comparator group, from an ethical point of view mainly, it may be appropriate to offer patient an invasive sham procedure. So the more invasive is the procedure, the harder it is to justify exposing patient in control group to risk that may be substantial and without unexpected benefits eventually. The learning curve, the result of a clinical experimentation on medical devices often depends on the expertise of physicians which are involved. So we have to consider that there is a period in which physician must be trained on how that medical device works and how to use it, and eventually all the problems connected to. So the different levels actually of surgeon experience and expertise may lead to different levels of performance when carried out intervention. So it's a very big issue in medical device experimentation. And here, just to give you a comparison between the two kinds of areas, we have, as you can see, in pharmaceutical clinical trials, we have a high standardization because, as I said before, they are divided in phase 1, 2, 3, eventually 4. And in medical device, instead, it depends on the clinical investigation plan. So the methods for pivotal trials. Here, in pharmaceuticals are highly standardized, so it means double-blind, randomized control. Instead, here, another one, it depends on the CIP. The irreversible effects on study subjects are rare. Instead, in a medical device experimentation, could be very common. And also, the research in vulnerable population is easier to detect in pharmaceutical clinical trials. Instead, here you have to need more accurate, a good checklist is needed. The companies involved eventually could be also startups, new startups in medical device experimentation. Instead, in pharmaceutical, we know there are mostly large companies which could conduct a good clinical experimentation. And the know-how is easier to build up in a clinical experimentation on pharmaceuticals because of dossier well experimented and eventually visualized then in different times, in different years of experimentation. Instead, here in a medical device, it's more difficult to build up a good know-how. And so thank you for your attention. And eventually, if there are questions, I'm here, or eventually for later we can meet and tell each other something. Thank you.
Giulia, are there any questions for Giulia? Giulia, in that case, I have a question.
Okay.
Obviously, the ISO standards can be somewhat difficult. And so if I was designing my photonic medical device, how many devices would I have to include in a typical trial at the early stages to make it equivalent to the pharmaceutical phase 1 trial? Is there any easy answer to this question?
Okay. It's not so easy because, I'm mainly involved in pharmaceuticals, so when you have to talk about medical devices, mainly Dr. Nuti is more expert than me, but eventually you can ask him for the ISO, which is very, let's say, specifically certified in discussing these kind of points. But your question is good because you have to, I think all your medical device you want to experiment must be included in a clinical experimentation at the early stages, because you have to design a good prototype, and you have to prove if that prototype could be harmful or good effectiveness, or eventually must be redesigned eventually. You know better than me because often medical devices are, let's say, a merge of different kind of small parts of a medical device, so of a bigger medical device. So in a clinical experimentation, it means you have to include all of them, all of parties. And if you have more than one, you have to put them all in phase 1, let's say, in an early feasibility study.
Okay.
Okay.
Are there any other questions for Giulia? If not, thank you very much again for your
lecture.
Thank you.